Good afternoon, everyone. This is my absolute pleasure today to present the view of the scientific committee on gene drive. And I would like to thank the organizer to invite me to give this presentation on behalf of the ARRIGE scientific committee. Unfortunately, it is rather late in Australia, so I am not able to take the questions in live. However, if you have any questions, please use the chat function, and I will be more than happy to reply to those tomorrow or later in the week. So really an important question that underpins our view is what is gene drives, how important it is, and why as ARRIGE scientific committee we have to come up with a position statement, more exactly a policy document regarding gene drive question. And that would be the main part of this presentation. So there are two instances that most of ARRIGE members know about gene drives. The first one is on deadly animals that are mosquitoes. These mosquitoes carry diseases like malaria, Zika, dengue, and kill a lot of people. More than for example, snake, lions, dogs also. The other instance is the emergence, the really widespread of invasive species. And why is it a problem? Is because it endangered native species and affect the entire ecosystem. So it's very, so this is those two instances where gene drive could play an important role, and where all the debate has been held for the last couple of years. And there are some examples there of issue that was brought regarding gene drives. So one is something you've seen quite a lot. If mosquitoes are so deadly, why not to kill all of them? And this is where gene drive comes in. The second instance is, for example, we can use gene drive to eradicate weak pathogen, for example, but for eradicating invasive species. And on the other side, this headline shows well that what was bought is gene drive too risky for field trial. So the question is where we are, what is the state of the technology, and what is the ethical issue attached to it, and what are the community engagement to make the technology better understood from everyone. So this is where I believe ARRIGE plays an important role. So before we start really digging into the gene drive question, I would have thought the basic would be to explain briefly what gene drives is. And I have this movie for you showing very well what gene drives is, and the example is on a mosquito there. So as you can see here, the classical mosquito gene technology is to replace the DNA. And then it makes the mosquito immune to malaria and breeds and spreads. Unfortunately, the spread is 50% inheritance, and as long as the generation comes, there are less and less GM mosquitoes. So that means we need to release a lot. So what gene drive is instead of what we do here is to insert the CRISPR cassette there, and the CRISPR cassette auto maintain and auto replicate. So what the consequence of this is, it really increase significantly the spread of the gene drive throughout the population. And as you can see here, that kills all the malaria parasites. And it spreads in 3 to 5 generations to the entire population. So this is the really important feature of gene drive technology using CRISPR-Cas technology. So the question is, what is the view of the scientific committee on gene drive? Why is it important? Because this gene drive technology has a strong potential, as just illustrated with the malaria parasite, and there are a series of issues that have been brought on it. So the first, before we go into the policy matters, what I would like to introduce is the process. How we came up with this position on gene drives. So the matter was raised from the ARRIGE board before the COVID lockdown in March, I believe, 2020. So what happened is it went to the ARRIGE Scientific Committee, so that I'm chairing, and what we came up to basically answer the simple question, what is our view on gene drive? We come up with a working group. So what this working group is part of the ARRIGE Scientific Committee with members that have worked on that question before. So they have deep insight of the gene drives technology. So the working group had a lot of discussion, and we drafted a position statement. And more state of the art really than the position statement. So what happened next? Once we have this draft, we went back to the ARRIGE Scientific Committee for peer review. And it was truly a peer review because it has significantly improved the quality of the document. And then we went for another layer of peer review from the board. And the document is was finalized, and you can read it now on the ARRIGE newsletter. So who were these persons on the working group? These are people that have worked previously on the question and that are largely involved with the gene drive technology. So I give you their role, their names, and it's all in alphabetic order. So the first one was Hakim Adegnika in Gabon, and Hakim has been quite a lot involved with the community aspects of the gene drive technology and in Africa. Secondly was Milan Bodbol. So Milan is a philosopher and ethics person, and she has been working quite a lot on reflecting on the ethical aspect of gene drive. Martina is based in Uruguay in South America, and she has been quite a lot involved in establishing the gene drive technology in South America. And as well, she's a lot involved with community aspects. The next one is Tony Nolan. So Tony has worked a lot in improving the gene drive technology for eradicating malaria, and has considerably improved the technology. The next one is Jerome Singh in South Africa. So Jerome is highly involved in the legal aspect, the ethical and community aspect in Africa. And he has played a pivotal role in establishing ethical framework for gene drive in Africa. Bruce Whitelaw has been quite a lot involved in gene drives in invasive species, and it gave us different aspects on gene drive besides just eradicating malaria mosquitoes. And the chair was someone external from the scientific committee for to avoid bias, and it was Christian Siatka from the School of the DNA in France. And I was part of this group as the scientific committee chair, and of course I have worked on the gene drive question as well. So as I mentioned, it has been quite a relatively long process, and as it is written there, we had quite a lot of Zoom meetings, a lot of email exchanges, and so on from the working group, and then with the scientific committee for the feedback. And it was very important feedback because it has considerably improved the quality of the draft. What I would like to emphasize, it was highly collaborative, highly open exchange on the questions, and we had conversation over 5 different time zones. So that means it is possible to have those conversations on Zoom despite all the COVID-19 lockdown and so. So the question is, what is the science, the state of the science on gene drives? So we come up with the view that gene drives is best suited for organisms with short generation time. And the one that comes into mind are the mosquitoes more than, for example, here the rabbits. And these are the most appropriate. And clearly to date, these are the organisms that show that gene drives have been the most successful. And there are currently 2 field trials going on. As I mentioned before, gene drives has been quite successful for these organisms, except for mammalian species with CRISPR it hasn't worked. I won't go, unfortunately, I don't have any time to go into the details, but suffice to say that there are alternatives other than CRISPR with gene drives, and indeed, gene drives have been existing for 30 years. So it's not all black and white, and while there are some field trials, there still are some series of technical issues need to be resolved. And one example is the resistance to the drive. So it's not, we are in a position where gene drive could be widely implemented. However, that gives us time to reflect and to come up with a strategy and a policy on the question. So it's not so bad. So on the regulation side, as most of you know, the regulation landscape is quite fragmented, as you can see here worldwide and here in this Africa map. And there are different types of regulation, from permissive, so for example in South America here, or in Africa, part of Africa, to heavily regulated like in Australia. So, however, there have been quite a lot of effort from WHO, the African Union, the CDC, Bill Gates, to devise a clear strategy, framework, and a legal framework for implementing gene drives. Because the regulation landscape is a lot fragmented, we thought the best to apprehend and to make it working is a regional approach. Why? Because if you, for example, release the gene drives here in this boundary, that would affect the other countries. So instead of trying to go for a global governance for now, so we would like to, but it's a bit challenging, we thought the best is to insist on a local regional regulation approach between multiple countries, and that would facilitate, and it's more realistic really, on the management and the ethical guidelines. As you would guess, the ethics question was the most debated between us, and as I believe it is still strongly debated. So what I would like to emphasize is it's all about responsible science. Beyond the utilitarian framework, benefit versus risk. And really, it's all about communication and clearly describe the shared risk and anticipate the benefits. Really, the most critical point is to communicate well and ethically. And why? Because the scientific knowledge is not equally distributed. So we made a distinction, north versus south difference. However, it is more subtle than this. All of this creates ideological tensions, and we need to think very carefully how to differentiate values and human interest. And I believe one important role that ARRIGE could have is on the community engagement. And it's really critical to provide, to consider the community's point of view, and a clear engagement and provision information is absolutely critical. And I would like again to emphasize this, that the community that is the most needed for gene drive technology are the most vulnerable. So we really need to take this into account. And the legal framework must capture this concern and community acceptance. And this is, for example, in the scientific committee, what Jerome Singh has been doing tirelessly. And the acceptance really depends on the good communication, participative and relation ethics discussion. And I cannot emphasize this enough. So what is the role of ARRIGE? What ARRIGE role could play? One is the explanation of concepts in the form of the democratization of knowledge. This is where ARRIGE sits in our view. And to promote the confidence in gene editing technology and taking into account all the complex and different aspects between ethical, cultural, and so of the gene drive technology. So really this is what we think, where the technology is, and where we think ARRIGE could go in that direction. So in conclusion, on the technical side, it seems to be the gene drives is getting more and more efficient. We have a better control, but we still a work in progress, and we are not ready for wide implementation yet. Something that I would emphasize this a lot is the CRISPR field is moving fast, and this shouldn't be underestimated. And the third point, and the most important one, is ethics and community engagement is the key for success implementation of this strategy. It's really critical, and I emphasize this again, limiting vulnerability, taking into account plurality of viewpoints is really critical. So this is our view. You can find this on the ARRIGE newsletter. And I would like to thank, on behalf of the ARRIGE Scientific Committee, all the people from the working group that have been working very well together, and the ARRIGE Scientific Committee, the rest of the ARRIGE Scientific Committee, for all the feedback on the question, and of course the ARRIGE board for the final feedback. Thanks.